Thank you for that very nice introduction. And thank you, Dr. Mance, for being here today. I appreciate that. Yes, I think I was one of in the early group to um, be a trip scholar, and I've enjoyed every minute of it. And it's really grown and advanced through the years. So I'm really happy about that. Now, can you see this okay,
Yes,
Stephen?
it's great.
All right.
Perfect.
So today will be a little different talk than I normally give. I'm going to kind of take you on the journey of how I went from when I first came here over 3 decades ago, and what we've been doing on this journey. That's been a lot of fun. A lot of people, I kind of joke about it, but I really mean it, that I find that research is fun. I've been able to be involved with a lot of exciting people in different fields, and I'm going to talk a little bit about that today. I'm going to just tell you about the journey here on this slide. My obligatory disclosure slide, just so you know, I do have some interactions with some companies. I will talk about a little bit of our startup company and then our licensing with another company. And then at the end, I'm going to conclude with some future directions that we're doing, where we're about to start, hopefully to start up another company here in the next few months. So I'm not going to go through the detailed science on this. If anyone's on the Zoom call today and is more interested, I'm happy to talk about it at any time. Just email me or get a hold of me. I'm more than happy to talk about it or send you publications. Stephen said you can find me pretty easily on online and on PubMed or through LinkedIn if you want some references. But what I'd like to talk about today is really this down here on the bottom is this protein that goes by two names, APE1, REF1 for apirin internuclease 1 and REF1 for redox effector factor 1. And I'm not going to go through the gory details on all the mechanism of what it does. But for today's talk, keep in mind that it has two major functions. One is in DNA damage and repair, kind of shown over here. And the other is this, what we call a redox function or redox signaling. And it's what's kept us working on this protein. And I was looking in preparation for this talk. I think it's on the slide, we made the first antibody to this protein in 1989. So we've been working on this protein, this target, a long time, trying to understand what it does. Over the years, it was originally found as a DNA repair protein. It fixed damaged DNA that happens spontaneously, normally, and after chemo. And then it has this other signaling protein that turns genes on and off. The latter function was found more in about 10, 15 years after the cloning of this gene. And it turned out that it's one and the same. So we kind of have it as the yin and yang. It does both the repair and the redox. And what's intriguing about that, and what's kind of led my career, is starting out in DNA repair. We've been doing a lot of work in cancer, and we're still doing that work. And we've been funded by the NCI to do that work. We've been looking at some of the roles of this protein in chemotherapy-induced peripheral neuropathy, which occurs when you're treated with a lot of chemo agents. You get peripheral neuropathy, and the role it plays in that. Recently, one of the drugs we developed, which I'll get to in a minute, was taken through my company and then licensed to another company to do a trial in diabetic retinopathy. So we've been looking in the eye disease. And then what our new adventure is going to be focused on is Crohn's and colitis, ulcerative colitis, IBD, where we have good data on our target protein and our drugs. Now, you're probably sitting there going like, how can this one thing do all these different things? And how can you focus on that? So what I always like to tell people is we never lose sight of this protein and what it does. It's just that once we found out some of the things it does, we then went on to develop compounds, different names. The one I'll talk a lot about today is called APX 3330. But this compound, which is an oral tablet, this compound binds to the protein and shuts off this function, the redox, but does not touch the repair. And that's an important concept on how we're able to look at this in different diseases. What's interesting and why we're looking at cancer and neuropathy and ocular disease and IBD is because it's the same protein and the drug's doing the same thing to the protein. It's just that when you start to look at the models of these disease in cancer, neuropathy, IBD, and ocular, a lot of the same pathways that are used in cancer are disrupted in these other diseases. In fact, in eye disease, wet age-related macular degeneration, some of you may know, some of you may even be treated, is that they use an injection in the eye called Avastin, which is blocks a normal growth of the blood vessels in the eye. Well, Avastin came out of cancer research. So there's kind of a theme here where a lot of times cancer research is leading to discoveries that are then used in other indications. And I think we're kind of right now, in my opinion, the poster child for that, because our protein in our drug we're using it in cancer, anti-neuropathy. We've done trials in ocular disease, and we're going to be doing it in IBD. So we're not unfocused. We're focused on our drugs blocking our one protein. It's just that the protein is involved in different cancers. And so I'd like to just take you on a little trajectory. I think today the talk is about translation. And I want to just tell you kind of the path we've taken. I'm also the co-leader of a program in the Cancer Center called Cancer Drug Discovery and Development Accelerator, CD3A, and we've recruited a lot of former Lilly folks to help move drugs along this pipeline. I'll show you. One of the things we like to say as our motto is drug development is not for the faint of heart, because it takes a long time. It can be very difficult, and you just have to be persistent. And I'm going to show you some of the roadblocks that you can run into. And so a little bit of history here. This is, I mentioned, when I was previously, I was at Loyola University Med School in Chicago, 87 to 93. And we were working on this protein, APE-REF1. I'll call it either name, APE or REF1 or both. And actually, it came out of work as I was trained. Actually, my PhD at LSU and then my postdoc at Rockefeller was in Drosophila research, but we were studying DNA repair in the Drosophila organism as a model organism. And eventually we started to move out of Drosophila into looking at mammalian cells and then eventually into humans and looking at DNA repair. And that's why it led us to this protein. And when I was up in Chicago, we developed the first antibody, which I like to say was actually the first antibody ever sold by Novus Biological. They licensed it from IU to sell it because I got tired of sending it out. And so the IU folks here got to deal with Novus, which has now been sold a few years ago. But we were actually their first antibody ever sold by them. And that's because the person who ran Novus was an IU alum. I always like to make sure we plug our IU alums who are doing business. And so Karen Paget contacted us and we did that. So I came here in 93. And then we've been working on the repair part here in the late 90s, early 2000s. And one day in, probably about 05, 06, John Bernard, who used to work at Lilly and founded Pearl Street Ventures, came to the campus where the dean at the time was Craig Brader. And he said, Dr. Brader, what, you know, anything going on? I'm looking to do maybe some investing in some companies and to help move things along from IU out into the clinic. And Craig very nicely pointed him toward me, and we started having discussions. And then in about 08, we founded Apexian. At the time, it was called Apex Therapeutics, right around 08 or so. Now, 08, 09. Now, that is, you can see, that is a wonderful time to start a company because the market crashed and everyone, all the venture funding, all the investors and everyone ran for the hills. And so we were able to get enough local funding, and I still thank John and his investors here, including BioCrossroads, Elevate, and others here in the state who came along and kind of helped us move it along, keep it going during these rough times after the market crash. What we were doing is we were developing new compounds and all. We got above the line is more of the company side of things, Apexium, and the bottom is more of the academic side, where we got grants to look on cancer. We started looking, using our drug in studies in the eye, which led to the trial, and working on different cancers, particularly pancreatic cancer and more recently some sarcomas and pediatric cancers. We renamed the company in 2015, and we were able to obtain additional funding. And in 16, we had an IND. That's what you need to file to be able to do a clinical trial. And in 2018, we started a phase 1 trial in cancer. Phase 1 trial is just a safety trial using our drug APX 3330. It was a trial where we ended up with 19 patients giving our drug, and we dose escalated to see the maximal dose tolerated. The trial was actually very successful. In fact, it took a year to do it. Usually a phase 1 trial is a few months. But because we had patients who, the trial had patients who had cancer, all different types of solid tumors. They had to have tumors that were progressing, where they had failed all other treatments, unfortunately, and that they weren't taking anything else at the time. So their tumors were growing. We found in about a third of the patients that we actually slowed the growth of the tumors, some over a year. So we were very excited about that data. Unfortunately, the tumors did not go away, but we slowed the growth. And so we were encouraged by that data that we hit the target. We have data now that we hit the target. We are just now need maybe a little more potent drug for the cancer work. What's interesting though, is because of the pathways that our protein and our drug targets in cancer. One of them is anti-angiogenesis, which is anti-blood vessel growth. The other is anti-inflammation. I think we all know about inflammation, and inflammation plays a role in a lot of diseases. Well, in the eye and retinal disease, particularly retina disease, inflammation and angiogenesis play a big role in a number of different diseases, including wet age-related macular degeneration and also diabetic retinopathy. But diabetic retinopathy, there are no treatments for it early on. And so what happens is the diabetes is causing it. You start to slowly lose sight over a period of time. And eventually, after a number of, it could be years or however length of time, they start getting these injections, like I mentioned, of this anti-VEGF compound. But there's nothing for early patients to have to stop the diabetic retinopathy from progressing, nothing to take. And so a company called Ocuphire Pharma and the CEO, Mina Such, approached us and said, look, we're studying your mechanism of action on your target. It's a novel target, hasn't been addressed in the eye, hadn't been addressed in cancer either. You're hitting pathways we're interested in, anti-blood vessel growth, anti-inflammation, and it's an oral tablet. So patients could take it as an oral tablet, not having to get an eye injection, because you don't want to start the eye injections until you have to. So, but this way, if you could take it along with your other diabetes medicine, take the tablet, and hopefully we would slow the progression. And so Apexion was able to do a licensing agreement with Ocuphire, and they started a phase 2 trial right during COVID, in the middle of all that, and were able to complete it and finally have the readout. It was completed in about a year, which was really a triumph on their part, because they had 100 patients, 50 got the drug, and 50 were placebo who did not. And then we able reported it out in the end of 2023. At the end of that trial was very interesting, because it was very interesting discussions with the FDA, because we were giving a tablet. So it's systemic, it's going all over. So you're affecting both eyes. And a lot of the trials that are done previously, when you do injections, you can have one eye control and one eye with the injection. You can't do that with an oral tablet. So the FDA, we would be the first oral drug ever approved, if it gets approved, for retina disease as an oral tablet. Most are injectables or local delivery. So we had a lot of good discussions with the FDA. They're trying to evaluate the data. And we had what's called an end of phase 2 meeting with the FDA. They approved our results and they said we could proceed to phase 3. And that's really where we are now with Occuphire. They recently changed their name to Opus. And there's the potential to do a phase 3 in diabetic retinopathy with an oral drug. While we were doing all of this, we had been doing studies with some colleagues in IBD. And how did we get into Crohn's and colitis? Well, we published our data. People saw our mechanism. They saw our work in cancer. They saw our preclinical work in the eye, and they saw the data from the clinical trial. The other thing I should mention, in both the cancer trials and the eye trials, the drug was perfectly safe. We really had minimal side effects. If you get high enough dose, you'll get a rash. You can move the dose down and the rash goes away. So this is very safe. And I think the reason is, and we're still trying to prove it, is because we're just blocking this function, which is kind of out of control in these diseases. But we're also seeing a protection of this function. It's still acting, which is very important. So we've seen neuronal protection. We've seen good positive things in normal cells, whereas in the cell that's out of control with increased inflammation, whatever, we kind of tamp down that and make it normal. So that's why we think it's a good target, good drug, and the side effect. Well, in Crohn's and ulcerative colitis, what are the things that cause this? Are inflammation. So once again, it's in our wheelhouse. It's the same thing. But what we found in a paper published with our collaborators, this work was actually done with a collaborator in Australia, Camilla Nogali. She's a wonderful collaborator. She has a great mouse model for colitis. It's actually a genetic spontaneous model. And what we saw when we gave 3330 in this model, we saw a reduction of inflammation and we saw neurons being protected. So if you have colitis, there's these neurons called enteric neurons that invade the gut, your gut, and help it for movement of food through the gut. And these get oxidative stress and damage and are very messed up in these diseases. Well, guess what we do? We're seeing an increase in the DNA repair with our drug while blocking this. So we're blocking the redox, blocking the inflammation, while we're increasing the repair to show the protection of the neurites. And we actually have a beautiful, it's her paper. She has a beautiful picture in her paper showing we see the neurons actually growing and recovering from the damage in this. So by doing our cancer work, and we still are, and looking at how it works, the mechanism of action, we're able to do the ocular disease because we had collaborators to work in the eye, and we're able to show that we're hitting pathways important with that. We had previously shown that we can protect dorsal root ganglion neurons from chemotherapy, which is an area we'd like to get back to, but we haven't been able to. But we did see neural protection, and guess what? Same thing we see in the IBD case, neural protection, reduced inflammation. So these all fit together in a nice pie, if you will. They're all related, really, by our protein in some ways. Let me make it clear, our protein doesn't correct and cure all of these. I'm just saying it's a target for these different diseases. And we can target this using our drugs that's very safe and move forward. And so what we plan to do right now, we're talking to people who are very interested in doing a spinoff from Apexion, a NUCO, where we would be focusing on Crohn's and colitis using our drugs and some other second generation of our drugs. And hopefully the goal, this is a goal, I'm just saying, within a year of now, we would be in clinical trials in IBD with an oral drug, because right now, most of the treatments in inflammatory bowel disease are biologics and they're injectable. They usually work for a couple of years, and then the patients unfortunately need to move on to something else. One of the drugs you've probably heard is big is Humira. It's a massive drug that recently came off patent. But we think ours is novel because it's a novel target. We block inflammation, we correct the neurons, and it's an oral tablet that the patients can take. Um, so that's really the gestalt of what I've been doing over the years. I've had lots of collaborators. We tend to keep our lab fairly small, but we have people we collaborate in cancer over the years, structure function. We recently, with Millie, we published a NMR paper showing our drugs binding. Millie's here on campus in biochem. We've published with Tim Corson recently with some postdocs from his lab, all the eye work, the preclinical eye work. And we have 2 new papers just coming out in that. Kumira has been fabulous working on the IBD work down in Australia. 2 chemists. Jim has been with my company, and Tim is part of the CD3A former Lilly folks who are collaborating with us. And again, we have folks here on campus in bioinformatics. Ruben, we have some interesting data in IBD. I could talk about some other time. Karen is doing in vivo, and then in my lab, and we've had a lot of funding from different institutes, the I, NIH, NCI, including my chair, the Tom and Julie Wood Family Foundation, Riley Children's Foundation, and others. And I've been fortunate. I like to say I'm just the mouthpiece up here. It's everyone else who's doing all the work. But it's been exciting because I've been able to go from Drosophila to cells to mice to humans to clinical trials to biotech startups. I've been able to work with people in cancer, in ocular diseases, retinal diseases, inflammatory bowel disease now, and anti-neuropathy. So it keeps me, I may not look it, but it keeps me young, and it keeps us very constantly thinking about other ways, because our goal is to get these things to the patients. You hear the phrase, the patients are waiting. That's what we feel about this. That's why we keep pushing, through a lot of different times, when times are good and times are bad, through recessions, through all sorts of things. People have stuck with us. And so we've been very excited that people actually have belief in our program. And I think they think there's enough data and belief that they're willing to maybe hopefully help us in financing and get this next program going in Crohn's and colitis, because the patients are definitely waiting there. And that's it as far as what I wanted to say. And I'd be happy to discuss with folks anything they want to talk about. Thank you for your time.
Thank you, Mark, for your time and for sharing that. That's a lot of information. And as you had talked about ahead of time, how do you present all that in a way that makes sense? What a beautiful way to share that with us. So this is the time we invite you all to come on screen and raise your hand in whatever way, or come off mute if you have a question or comment or some idea for discussion. So I'll pause for a moment, see if there's anybody that would like to say something.
I've got a question for you. As you said, many diseases involve inflammation. So do you see this going to treat other diseases as well?
Yeah, thanks for that. I would love to. I'm always open for collaborations. We have our drugs. We hand them out. So yes, I could see there's a number of areas. I mean, we've looked at sepsis a little bit. I haven't gotten too deep in that. We've thought about other, anything that is an inflammation involved in it, I think is the potential. One of the interesting things about, like in the diabetic retinopathy trial, the phase 3, going forward, that we'll probably be looking also at, will we have an impact on diabetes, right? Because that's actually an inflammation driven, and look at, by taking a pill systemically, will you be able to correct the eye or protect the eye and do that. So no, that's how we got involved in the eye and got involved in colitis and stuff, because we had investigators who read the literature and then contacted us. So if you have any ideas, we're happy to do this. A lot of people mention to me skin diseases, right? Rashes and various things, and even in animals, right? I mean, I think that there could be some dermatitis, some various areas that I think could be potential interest. It comes down to bandwidth issues. But if someone collaborates and wants compounds and reagents, we're happy to share. We love doing that. Learn something new every day. Thanks for the question.
Mark, one of the striking things as a lay person was the juxtaposition or coming together of the yin and yang of two different names having such different impacts simultaneously. And I'm sort of curious as a scientist, when did that light come on? How in the world did you manage to bring these two things together, since they were clearly living separate lives?
Yeah, no, that's great. So I cut my teeth on DNA repair. So I was working on it as an ape protein, aporinic nuclease, the name. We were studying it and just studying all the repair parts in the early 90s. But in about the mid 90s, two things happened. One, there was a researcher at St. Jude's, Tom Curran, who was actually working on transcription factors, the proteins that turn genes on. And he was studying that there's a protein that seemed to interact with these proteins and turn them on, talk to them, do something to them, and that's the redox cycling, and get them to turn on. And when he cloned the protein, and this is for some of us, and this is back in the old days where you went in the cold room and purified proteins day and night and ran columns, and all before the sequencing, everything could now be done in hours. They purified this protein and they followed this characteristic of its redox activity. And when they finally, within a couple of years, got it finally cloned and figured out what it was in the sequence, guess what? It was APE1. They were one and the same. So they kind of discovered it and studied it. We saw that and then started looking at both the repair and redox. You can mutate the repair and the redox function is still active. You can mutate the redox and the repair is action. So then we started going after drugs. We're still trying to get drugs that block the repair and block the redox. One quick note I have to say is years ago with Millie over here in biochem, we wrote a grant, and some of the data in the grant, we had cloned the zebrafish ape ref1, if you will. Zebrafish, which, vertebrate but not a mammal, they have the DNA repair part of the protein. But there's one particular amino acid, cysteine, that is missing. That's the active part of this signaling. And that's missing in the zebrafish. And so the zebrafish had repair, but not the redox. We mutated because she does crystallography. And so we crystallized it and saw it was in the same place as the cysteine. We changed the amino acid there to a cysteine, and zebrafish, lo and behold, turned into a redox and a repair protein. So this is a late evolutionary event. That's a gain of function where you went from just repair, and the repair, I should say, is conserved from bacteria to humans. But all of a sudden, at the zebrafish level, we can make the zebrafish human or mammal, and all of a sudden it gained function. It's one of my favorite experiments. We submitted a grant, they hated it, did not get funded, got the paper rejected 2 or 3 times. We finally got it published. And now everyone talks about it because they're like, well, that was great because you had a natural model for how this evolved and stuff. So, but thanks for asking that, because so that's how they came together. It's really different fields, and we've been working on it. Sorry for the long answer.
No, that's a good story. It really is. And the other thing, just as a 70-year-old with arthritis, inflammation. You say inflammation, I think arthritis. My thumb just reminds me regularly.
Yeah. Yeah. Like I said, we have, anyone who has models that makes sense, we're willing to look at. And the beauty is the drug the lead drug is so safe. That's the key. As an oral drug, it's so safe.
Yeah, it's not only safe, it has a positive impact. They've
Yes,
done
that's always good
exactly-
too. Yes,
Yeah.
right.
Wow.
Other thoughts for discussion from our audience?
Mark, hey, it's Dave Boyle here.
Hey, Dave.
How are you? Hey,
Good. How are you?
congratulations. Wonderful
thanks.
career, buddy.
Oh, thanks.
So my question is pretty naive, I think, but maybe profound. So this drug is so safe. We know that the redox state of cells is so important in the function of cells all over the body, whether they're in the GI tract or in the brain or where. And yet your drug is safe. And I would think that it would be difficult to just target the diseased cells without affecting all the rest of the cells in the body. So I don't know if you could comment on that further.
Yeah. No, that's good. That's what all the investors and people said. This drug, you can't take it because it's going to blow up all your other normal cells. But I didn't explain it detailed enough. So the protein APRA1 is talking to the transcription factors of protein-protein interaction, and our drug blocks that from occurring. So it's not a general redox, Dave. It's not like we're changing the glutathione levels or thyroidoxin, and it's not a general redox. It's a redox signaling between proteins. So that's why it's specific. And what we've found, thanks for asking the question, actually. What we have found is what happens is the normal cells are in the cancer cells or in the eye cells or in the colitis, the cells that are not functioning correctly. They have elevated oxidative stress and have elevated inflammation. What we see in all cases, and we don't know why this happens, but what our drug does is dampen the activity down. It's like calming the storm, right? It brings it down to normal, but we don't go below normal. We don't go down to zero. We're just bringing everything back down to normal. So in normal cells, we don't have a negative effect. What we do see is in the normal cells that are in oxidative stress, because by blocking the redox side, we see it's probably because of the way the drug works and unfolds the protein, we actually see a stimulation of positive repair. So that's why it's probably safe. A literal hand waving here on that part, but we're working on that. But we do know, but to answer your question is the bottom line is we never drop anything really below steady state levels. So we call it calming the storm, really.
Great. Thanks, Mark.
Yep. Thanks. A good question.
So Mark, along that line, I'm wondering about, as you've talked about this is a remarkable path and the things that have happened, but you said very clearly, this wasn't just you, it's like a whole bunch of people. So what are the opportunities for, like, how do you see this working for early career folks or students that are like coming in? How does that weave into how you make all this happen?
Yeah, that's actually a long answer, and I'll try to not make it long. In academia, I let me say it this way. I think things to start to do this translationally, but startups licensing with IU and their system now is really so much better than
Yeah.
when I started, to be fair.
Yes.
And there's a lot of scars I have and stuff, which are great that I've learned from, that I think now you don't have to go through. And for example, in the
Thank you
CD3,
for forging the path. We'll say that.
thank you. Yeah, you're welcome. But you do learn by that way. But we now, like in our programs with all the junior infection, and I'm going to talk about cancer because we do that a lot, as associate director in the cancer center, is we meet with the young faculty. We try to see what they're working on. We help them learn when they should file their intellectual property, when not to. We help them see if they have a target, where it would go. Is it really rational to move forward? And we're not a drug company. So it's not about making a drug all the time. It's about moving their science, from discovery along the path so that it becomes attractive to someone who might want to license it or do a startup or whatever. Whatever path it takes to get it to patients who need it. That's the key. And so I think the system is much better set up. I think there's much better mentoring. The idea is mentoring, as we all know, right?
Mhm.
Everyone probably on this call knows that that's the key to all of this. And so in our case, in cancer, with people who have targets, that we set up teams, we meet with them regularly, and we help to do things, get things done for them so they don't have to spend their time, particularly for junior faculty, young faculty, because they have to focus on their academic career, papers, pubs, grants, everything. So we try to take a lot of the burden for this development and moving something along so they don't have to worry about, how do I get this done? How do I get that done? We do it for them. We plot out a critical path for moving things along. And we have, in the cancer side, for example, we have 15 former Lilly, some and non-Lilly, people who have over 300 years of pharma experience. So we use that experience around, here's what you have to do. And there's certain things you just have to do. If you're going to move along, you like it or not, this is what you have to do. The FDA is going to want to see this, period. And so if you know what to do and not waste time on things you don't need to do, that is huge. It's kind of, so I don't know if that answered your question at all.
It does, yes. So what I hear from that is, for early career folks, is, you said it, mentoring, that you get connected, and to take advantage of the wisdom of those before you, to know like where to spend your time. The other thing I hear you say is that it's about relationship. I always have to bring that in because I'm the social science guy, but it's about relationships and how that's been important for you. Not only in academia, but how you forge that outside of the university, of the academe. Talk about that.
Yeah, that's a good question. When I first came here, about the second year I was here, there was a pediatrician in oncology, Phil Brightfield, and he and I had a cup of coffee. And Phil said to me, he said, "Mark, there are a lot of really bright people, in science, but you want to work with bright people who are easy to work with."
Mm.
And I've kind of taken that to heart, in the sense that um, I've worked with both on the company side and in academia, and is that you collaborate with people, and if it starts out difficult, it never gets better. I've been lucky. I think when we started our company, we had a lot of people who actually probably looked out for my interests as much as theirs. And then that's kind of spilled over into some of our interactions with, and hopefully with this new company we're starting, which includes some of the people we've had before. Same thing in academia. I've been fortunate to work with senior and junior people who are fun to work with, who are on the same goal. That's what I would just tell people, is that, it's just not worth it. I think when I was here, I had 1.5 bad collaborations. It was painful, and I cut it off, and things were fine. I just think it's very important to work with people that you want to work with. I think that's
Yeah,
probably
so-
life
and-
too, right? But
Well, yes, and having a cup of coffee and getting to know each other and to build those connections. That is a piece of this. It makes some sense. Yeah.
absolutely.
Yeah.
Yep. Well, remember that also. Remember that book, Good to Great? It's
Mhm.
pretty old now, right? One of the things that I remember, actually, I remember talking to Ora Peskovitz about this when she was here, and one of the things I took away from that is the bus, your team. It's getting the right people on the bus, but the wrong people off the bus.
Mhm.
And I think a lot of times, I'll just say this, I think a lot of times we're good at getting the right people on, but not as good at getting the wrong people off the bus. And I have to say that I think that's very, very important, because you want that bus to work smoothly, and you need to do that. So
Well, I remember hearing Ora Peskovitz talks one time about complex environments, and that what she ascribed to was saying yes to opportunities as opposed to just saying no to things, which really resonated with me because I'm one of those people. But I think you are too. When you say yes to things, that's how you find connections and things that you didn't know. But and if it doesn't work, it doesn't work. But at least you've made those opportunities. So maybe
Yes,
that's funny you brought her up in that way. Yeah.
yes. Still in contact with Aura. Yep.
Yeah.
Aura, actually, I have to say, I was her first hire when
Oh,
she was
wow.
head of pediatric endocrinology. She hired me here from Loyola.
Wow. I'm glad we got you. Yeah. Well, are there other comments before we begin to wrap up this official time with Mark today? Any other ideas, comments, collaborations? Well, then let us take this moment to officially thank Dr. Kelley for coming on today and sharing. You are encouraged to go to the TRIP website. It's trip.indianapolis.iu.edu and go to the Scholar of the Month section so you can read all about his work. There's a wonderful Q&A section there, which gives a little more insight to Mark and his work and what he thinks about it. So we encourage you to take advantage of that. Reminder that we'll follow up at some point. It's the holiday, so we won't do it right away, but we'll follow up with some information and a link to the video if you have others that maybe would like to see this. But we can't thank you enough, Mark, for coming with us today, sharing about this, being a really good model about what we see as translational research, about taking ideas and putting them to practice and then seeing where it goes and being so open about that. So thank you. There's some
Thanks.
applause that had come around, but thank you for your for joining us today.
No, thanks so much. This has been fun. I love to talk about this. So this is great.
Yes, we know you do. So we'll officially end here. And but if folks would like to stay on for a few minutes, we always hang out, but we know we like to end officially. So those that need to get off to their next appointments can do so. But we'll be here for just a few more minutes to have the follow-up conversation if you'd like to stay. So thank you all for coming.